speak in turns. They're both from colleagues of mine from the Institute of Cancer Research, and I'm sure Trevor won't mind me saying he's actually a mathematician by background. And what they taught me over the last few years within our Centre of Translational Immunotherapy is enormous, and I think reflects the bits I was saying at the beginning about we generate all this data, how do we understand it, how do we apply terms like artificial intelligence, machine learning, and that was the other part, myself and Sarah, the clinicians, if you like, and more at the biological end. But hopefully what Trevor and Angaraj can tell you a bit about is how we need to all join together from very different angles to really make the most of what we're learning. Trevor.
Thanks. Well, hello. It's a real pleasure to meet you all this evening, and I'm extremely grateful for your support for our centre at the ICR. It makes a tremendous difference. So I lead a laboratory which is called the Genomics and Evolutionary Dynamics Lab, which is quite a handful. So for short, we call it the Jedi Lab, because it sounds cool, but also because I love Star Wars. And I think my love of Star Wars is actually this deeper love of sci-fi and a vision of the future, which is why I also really love AI and the possibilities that AI offers us in cancer research for having a different vision about what the future might look like. And I want to, talk to you a little bit about that today. So how are we starting to use AI to tackle our understanding of cancer? And so what I want to say actually follows very nicely on from the fantastic talk from Sarah about trying to understand how cancers evolve. And so what we mean by cancers evolve is that they change over time. As you've heard, they grow in our bodies for many, many years, often decades prior to us knowing that they're even there. And through that long, protracted growth, they change a lot, as Sarah so eloquently explained. And we really want to get a handle on that long process of how these cancers change over time. We also want to understand how they change when we treat them. The reasons that some of our treatments stop working, virtually all treatments work initially, but then after a period of time they may stop working, is because the treatment forces the cancer to change. It forces the cancer to evolve. And we want to understand that process of change, that process of evolution, in order to be able to use the treatments that we have better. And so what we've been trying to do in our lab and in the Centre for Translational Immunotherapy, the CTI, is try and make ways to read the secret diary, which I think is written inside every single cancer in some cryptic form. So you're all very familiar with a picture like this. This is, of course, the rings in a tree. And as you all know, you can count how many rings there are and work out how old that tree was at the time it was cut. You can also imagine that you might be able to take a core sample from each of these tree rings and run some analysis in a lab of that sample of the core of the tree ring and learn about the environment the tree was in at that time. There's a secret diary telling you about the history of how a tree grew written in its rings. And much in the same way, there's a secret diary that's written in all of the different molecular and genetic changes that exist within a cancer, that if we know how to read, like we can read tree rings, then we'll be able to understand how those cancers have changed and evolved over time, and also how they change and evolve through the treatments that we give. And it's that understanding of change that gives us hope to be able to deal with cancer better. So here's an example of what the tree rings in cancer look like. This is a picture taken in our lab of this is from a bowel cancer, and it's a very thin slice of a bowel cancer that we'd call a section. And what we've done is stain using a machine for lots of different types of cells that you can see exist in this cancer. So the different types of cells are in different colours. I find this picture really interesting. Cancer is obviously a horrible thing, but I think there's a kind of strange beauty in this picture as well about how these cells are organised and the patterns and the shapes that they take. So here the purple cells on the bottom and the left side of the picture are the cancer cells. And importantly, these yellowy cells and the big patch of red cells in the kind of centre top of the picture, they are the immune system. And so you can see this complex environment that exists, this ecosystem, if you like, of the cancer cells and their interplay with all of the different immune cells that exist in this cancer. You know, there's immense complexity in this image that you can all appreciate just from seeing the picture. And this complexity is where we try and use the AI to make sense of this. There are all kinds of patterns in here, all kinds of relationships that define how these different types of cells interact and where they are and how they move around over time. And that's where we're trying to use the power of AI to find those patterns and also to learn the processes that generate those patterns so that we can better understand how cancers grow. Another kind of data that we make a lot of, we do a lot of work using AI to understand is cancer genome data. So as Sarah explained, there are mutations that happen in the cells in our body, and some of those mutations make cancers grow in the first place. So on the left here, you can see a depiction of a cancer genome. So each of those wedges in this circle represents one of the different chromosomes, the strands of DNA that exist in a single cell. And these different colours, all this kind of complexity you see below represents different types of mutations that change that DNA sequence in different ways in a single cancer cell. And so there's all this kind of complexity you can see in that drawing there that represents the many changes in the genome that have typically happened in a cancer cell. Again, there's tremendous complexity there, and it's the power of AI to spot patterns and also to learn the processes that generate those patterns in the first place that offers us a new way to understand these data. So the kind of things we want to be able to do are to be able to have a picture like this here. This is a graph where we have time tracking the tumour's history on the x-axis here along the bottom, and on the y-axis here up and down we have the size of different populations of cells that exist within a cancer. So here there is a red population and a blue population that you can see. If you can imagine a hypothetical situation where we have two drugs, a red drug, a drug for the red cells, or a blue drug, a drug for the blue cells, and we have to decide which drug to give. Perhaps we can't give both together because it's too toxic for a patient. You might imagine that if we measure at this point in time, then the red population is very dominant, so we think, oh yes, we need to give the red pill, that would be the best treatment for this tumour. But actually, if we were able to get a window into the future and know what's coming, that red population is on the way out, and actually the blue one is about to take over, and that blue one is very minor at the time we actually measure. And so really the correct choice here would be to give the blue pill, not the red pill. And so being able to take the data that we can have today, use AI to understand the processes that generate the patterns in those data, and then we can push forward, we can make a forecast about what the future is likely to hold and make decisions based on that anticipation of what the future's likely to be like. And so the vision we have is that with the power of AI, we can take all of this data we have and make treating cancer like playing a game of chess. We can anticipate how the cancer will change given the move that we make with the treatment that we have today, and then be ready and waiting with our next move to respond to that and to have more effective treatments. And this same paradigm of playing chess, as Sarah so nicely explained, also works in a preventative setting. If we know there's these precancers there, and if we perturb them in some way, say with a vaccine, they may change. And knowing what those changes are mean that we can very carefully tailor our preventative strategies to always stay one step ahead. And so it's this kind of personalised approach of knowing how an individual cancer will change that Angaraj is a theme that Angaraj is going to pick up on and speak more about. So I think maybe we should take questions together at the end. Do you think so? Yeah, let's do that. Yeah.
Right. Thank you, everyone, and first of all, I'd like to thank CRIS Cancer for this opportunity, and also Alan. So, I mean, the previous speakers obviously made my life easy. So, just to tell them on my lab, I mean, I also initiated the Center for Global Oncology. The whole idea is to eradicate disparity in cancer research as well as treatment. However, we contribute to the Center for Tumor Immunotherapy with the Data Science Center and see how we could actually improve response to immunotherapy for various cancers. So again, as I mentioned, the three previous talks made my life easy with 4 keywords here. One, obviously the type of treatments, which I think I don't have to introduce again. The second thing is biomarker. Third thing is personalized medicine, and obviously AI. So what I'm going to do in next few minutes is going to tell you that how AI could really do things which probably we haven't imagined, and that's the power we are seeing currently in our day-to-day life. And can this be extended to the